The topic of lecture number 10 is epidemiological significance of parasitic representatives of the flatworm type, biology, pathogenical action, diagnosis, and prevention of trematode and testoda classes, cystic and alveolar echinococcosis. The purpose of the lecture is the study of the morphology and biology of cystic and alveolar echinococcosis. The lesson plan is the echinococcosis, cystic echinococcosis, the echinococcosis carnosus sensurato, alveolar echinococcosis multilecularis. Echinococcus is a genus within the Stoda parasitic class of Platyhelminthes. Is a phylum colloquially known as flatworms. Even at the dawn of Western medicine, this cirrhosis disease with characteristic fluid cystic lesions had already been recognized as ancient Greek physicians such as Hippocrates, the Epictetus, and Galen. Echinococcosis is a zoonotic infection caused by adult or larva, often called metacystosis stages of cystosis belonging to the genus Echinococcosis and family Tenidae. These organisms live in the intestines of the defined horses as adults, and in the internal organs of intermediate horses as cyst-like larval stages. Some species of Echinococcus circulated as domestic animals, also occur in wildlife, often called salvitic cycles, or have both domestic and salvitic cycles. Adult tapeworms are usually carried asymptomatically in their defined horses, which can include dogs, cats, and with carnivores. Carnivorous dogs are particularly important in zoonotic transmission due to their defecation habits and close relationships with humans. Many mammals, marsupials, and including humans, livestock, and pets, wildlife connect as intermediate hosts. Intermediate hosts are usually asymptomatic in the initial stages, but the growing larval stages, which often develop in the liver, the lungs, may eventually cause illness and sometimes death. Echinococcosis is a major public health problem in some countries, and it may be imaging and reimaging in some areas. Cystic echinococcosis is the most common form of the disease, is caused by members of the Echinococcus carnosus sensulato complex, because larvae of these organisms usually develop and discrete single cysts. It is least severe and the most treatable form of the Echinococcus multilecularis, often called alveolar echinococcosis, and Echinococcus vocally, often called the polycystic echinococcosis, are more serious and difficult to treat. The latter organisms can proliferate in intermediate hosts, forming masses that can infiltrate into organs and may disseminate to distant sites, including the brain. Echinococcosis is a disease that has been recognized by humans for centuries. That has been a mention of it in the Talmud. It was also recognized in ancient scholars such as Hippocrates and Eretheus and Galen and Rhazes. Although echinococcosis has been well known for the past 2000 years, it was not until the past couple hundred years that real progress was made in determining describing its parasitic origin. The first step towards figuring out the cause of the echinococcosis occurred during the 17th century, when Francesco already illustrated that the hypothesis of Echinococcosis were the animal origin. Then, in 1766, Pierre Simon Pallas predicted that these hypotheses found in the infected humans were actually larval stages of tapeworms. Tapeworms, a few decades afterwards, the 1782 goes accurately described cystis and tapeworm heads, while in 1786, Echinococcosis granulosus was accurately described by the batch. Half a century later, during 1850s, Corban Thibault showed through a serious experiment that Echinococcosis cystis do cause adult tapeworms in dogs. Shortly after this, in 1863, Echinococcosis multirecularis was identified by Rudolf Lockhart. Then, during the early mid-19th, the more distinct features in Echinococcosis granulosus and Echinococcosis multirecularis, their life cycles, and how they, because disease were more fully described as more and more people began researching and performing experiments and studies, while the Echinococcosis granulosus and Echinococcosis multirecularis were both linked to human Echinococcosis before the shortly after 20th century. It was not until mid 19th what the Echinococcosis oligarchus and Echinococcosis fuguli were identified as shown as being causes human Echinococcosis. Taxonomy is the phylogenetic tree has been created for several species in this genus: Echinococcosis oligarchus, Echinococcosis fuguli, Echinococcosis multirecularis, Echinococcosis shaccius, Echinococcosis equinis, Echinococcosis ortlepi, Echinococcosis granulosus. First diverging species are neotropical endemic species: Echinococcosis oligarchus and Echinococcosis fuguli. The Echinococcosis ortlepi and Echinococcosis canadensis are sister species as Echinococcosis multirecularis, Echinococcosis shaccius, Echinococcosis canadensis, and is related to Echinococcosis granulosus. The clinical forms of Echinococcosis, the metastasis of all four recognized Echinococcosis species can infect humans and causes various forms of Echinococcosis. Among these forms, Cystic and Alveolar Echinococcosis are of special medical importance. Cystic Echinococcosis is the causative agent of Echinococcosis granulosus. The diagnosis often called hydatitis and hydatidosis. Alveolar echinococcosis is a causative agent of Echinococcosis multirecularis. Multirecularis and causes alveolar hydatitis in humans. Polycystic echinococcosis, causative agent of Echinococcosis vogeli. This causes Echinococcosis vogeli echinococcosis. Unitistic echinococcosis is a causative agent of Echinococcosis legatus echinococcosis. The two most important forms which are of medical and public health relevance in humans are cystic echinococcosis and alveolar echinococcosis. Alveolar echinococcosis etiology is echinococcosis is the caused members of genus Echinococcosis parasitic tapeworm in the family Tenidae and the subclass Testoda. Testoda larval infections in the intermediate hosts are also called hydatitis. Species known to affect humans or domestic animals include Echinococcosis multirecularis, the members of the Echinococcosis granulosus, sensorator complex, and Echinococcosis shaccius and Echinococcosis vogeli. Echinococcosis oligarchus. The name of the last organism can differ between publications. While it has been called Echinococcosis oligarchus since the 1920s, some authors now argue that was based on misspelling and inconsistent with the naming practices, but it should be called Echinococcosis oligarta. Until the last 10 years, the Echinococcosis granulosus sensorator complex was considered to be a single species divided into 9 or 10 strains of genotypes. genotypes. Echinococcosis larvae develop differentially in the intermediate host, resulting in disease with different clinical causes. This disease caused by Echinococcosis multilecularis is called Alveolar echinococcosis. Cystic echinococcosis is caused by the numbers of the Echinococcosis granulosus and solar complex. Echinococcosis alligatus and the Echinococcosis voculi causes various echinococcosis is often called neurotropical echinococcosis is based on their larval forms in the usual intermediate hosts. Was the usual use term Cystic echinococcosis for Echinococcosis alligatus and for Cystic echinococcosis for Echinococcosis voculi after forms of the larva seen to date and the human cases. Echinococcosis is a cycles between on and more defined and intermediate hosts. Intermediate hosts typically in predator prey or scavenger prey cycle involving mammals and marsupials. In some intermediate hosts, larvae tend to form non-infectious, often called sterile, rather than infectious, often called fertile cystis. These hosts, which may also be called accidental and aberrant hosts, can become ill but do not contribute significantly to the organism's life cycle, whereas a host is a two intermediate host and an aberrant host is not clear in many cases, especially when only a few animals have been investigated. Carnivores, especially canids, felids, mastelids, and hyenids, usually act as definitive hosts. Echinococcosis cycles between on more definitive and intermediate hosts, typically the predatory prey and the scavenger prey cycle. Predator cycle as 20 Echinococcosis granulosus, sensus strictor, Echinococcosis canadensis, Echinococcosis outlepi, Echinococcosis multilecularis, Echinococcosis voculi, and the Echinococcosis alligatus have been detected in people. Through Echinococcosis alligatus seems to be very rare. There are no reports of humans infectious with Echinococcosis echinuens and Echinococcosis felides to date. One organism found in a person in Africa appeared to be related to Echinococcosis granulosus and strictor, Echinococcosis felides, but couldn't to be assigned to any of the known genotypes of Echinococcosis granulosus and so lato, and might be an unknown wildlife strain. Echinococcosis tapeworms have an indirect life cycle which can only be completed with both as intermediate and defined host, typically predatory or scanviginal, and its prey. Carnivores, canids, and felids are served as defined hosts for the adult tapeworms. They are herbivorous prey. Ungulators, rodents, and lagomorphs act as intermediate causes for the metacystosis. Humans are generally not directly involved in the transmission of cystic echinococcosis or alveolar echinococcosis, although under certain unique and unusual circumstances, such as reported in the Turkana region of the Kenya, humans act as intermediate host for Echinococcosis granulosus infection. The life cycles of the echinococcosis are dependent of the predatory prey associations involving two mammalian hosts. Definitive hosts act as carnivores. Intermediate hosts act as humans, domestic animals, ungulators, rodents, and lagomorphs. Hundreds to thousands of 3 to 7 millimeters long echinococcosis adult worms develop in the intestines of the definitive hosts. The last segment or proglottids, each worm matures to produce eggs that are released in carnivorous fields into the external environment. In turn, humans or The intermediate hosts ingested the eggs, which hatch in intestine to release oncospheres that pass through the portal and lymphatic vessels and reach to the liver, where they usually settle and develop as larval stages, often called metastasis or hydatitis. Less frequently, they may also reach the lungs, brain, bones, and any other organs of the human or intermediate hosts. Portoscolysis is the fertile forms of the parasite produced asexually by the metastasis are released into the hydatid fluid when ingested by defined hosts. Portoscolysis evaginate. The scolysis are aided by the bile salts, and after attaching to the intestinal wall, they develop into the mature egg-producing adult worms, and the life cycle is continuous. Epidemiology and transmission. Echinococcosis granulosus is a sensory complex occurs worldwide. With a few exceptions, such as Iceland and Greenland. Its distribution tends to be focal. Human illness are particularly common in parts of South America and the Mediterranean basin, Eastern Europe and Asia. Of the members of the complex, Echinococcosis granulosus, Echinococcosis canadensis, and Echinococcosis ortholepy are cosmopolitan and widespread. There are scattered reports to the Echinococcosis equinus in parts of Europe and the Middle East and Africa, and it probably also occurs elsewhere. Echinococcosis multilecularis has been reported in much and northern and central Eurasia, eastward to Japan. It's widely distributed in continental Europe. Echinococcosis tuberculosis, Echinococcosis legatus have been found in Central and South America. Echinococcosis schiaquisius has only been reported from the Tibetan plateau in China. A number of herbivorous and omnivorous animals act as intermediate hosts of Echinococcosis, and carnivorous act as definitive hosts. The pattern of distribution for the cystic echinococcosis has remained essentially unchanged of the past 2 decades, with the areas of the highly endemicity including Western China, Central Asia, and South America, Mediterranean countries, and East Africa, and the main risk factors being contact with dogs and raising livestock. 5,000 new cystic echinococcosis cases are still diagnosed annually, and in Argentina, Brazil, and Chile, Peru, and Uruguay. Alveolar echinococcosis has been public health concern in northern Japan for the past 40 years. Mass screening with ultrasound and serology in China have confirmed a high incidence of alveolar echinococcosis on the Tibetan plateau. Alveolar echinococcosis is also endemic in Central Asia, with high endemicity both of Echinococcosis multilecularis, Echinococcosis granulosus in Kazakhstan and Kyrgyzstan. Alveolar echinococcosis incidence has doubled in the previously recognized areas endemicity France, Switzerland, Germany, and Austria. In regard to North America, then North Central United States, Northern Alaska, and northwestern Canada have long been areas of Echinococcosis multilecularis endemicity, but the parasite geographic range appears to be expanding due, at least in part, to increased and improved sampling efforts. Targeting of defined host was than foxes. Other foxes current recognition species within the genus Echinococcosis and their preferential hosts and the geographic distribution. Distribution presents in tables and tables in presence in tables. Echinococcosis granulosus sensu structure complex is a defined host carnivorous. Intermediate hosts is domestic animals. Domestic animals in domestic animals is a human cases reported in many countries. This distribution is cosmopolitan. Echinococcosis multilecularis is a defined host is wild carnivores, including wolves and the foxes and the domestic the dogs and cats. Is intermediate hosts. Intermediate hosts is rodents. Rodents is human cases reported in many countries. Many countries is distributed in Eurasia and the North American countries. Cystic echinococcosis is a causative agent of echinococcosis granulosus sensulator hydatidosis or cystic echinococcosis is a zoonotic disease caused by parasite Echinococcosis granulosus, which form of the taxonomic perspective is currently considered a multi-species complex referred to as Echinococcosis granulosus sensulator. The species identified in this complex are Echinococcosis granulosus sensu strictor. Genotypes is G1, G2, G3. Equinus genotype is G4. Echinococcosis ortholepis genotype G5. Echinococcosis canadensis genotype G6, G7, G8, G9, G10. Echinococcosis felides is a lion strain. Echinococcosis granulosus sensu strictor, particular genotype G1, is the most widely distributed species at a global level and accounts for approximately 80% of human cases as a hydatidosis. Common name unilocular hydatid worm. Hydatid worm. The hydatid disease caused by granulosis is prevalent in most parts of the world and is the most extensive in sheep and cattle rearing countries, Australia, Africa, and the South American countries. South American countries is Echinococcosis granulosus strains, including G1 to G10 strains. G10 strains is G1 is was present in our country. Our country, including is Europe, Middle East, Africa, Iran, and India, Nepal, China, Russia, Australian mainland, Tasmania, New Zealand, United States, South America, and Sardinia. The defined because the dog, fox, indigo, jackal, and hyena. Intermediate hosts the sheep, cattle, pig, camel, goat, macropods, wild boar, and reindeer. The effective for humans is the Echinococcosis granulosus infection is infective for humans. The causes is worm has a life cycle that requires defined hosts and intermediate hosts. Defined hosts are normally carnivorous, such as dogs, while intermediate hosts are usually herbivorous, such as sheep and cattle. Humans function as accidental hosts because they are usually dead and for the parasitic infection cycle. Is Echinococcosis granulosus defined hosts dogs and other canidae. Intermediate hosts is domestic animals, including wild herbivores. Geographical distribution. Echinococcosis granulosus sensulator occurs particularly worldwide and more frequently in rural grazing areas where the dogs ingest organs from the infected animals. The geographical distribution of individual Echinococcosis granulosus genotypes is variable and area ongoing research. The lack accurate case reporting and the genotyping currently prevents and precise mapping of the two epidemiological pictures picture. Transmission, as one can see from the life cycle illustrated above, the all disease-causing species Echinococcosis transmitted to intermediate host via the ingestion of the eggs and transmitted to definitive hosts by means eating infected is containing organs. Humans are accidental intermediate hosts that become infected by handling soil, dirt, animal hair that contain eggs. Exhibited, the adult worm lives in small intestine of dogs and other canine carnivora, including wolves and foxes. The larval stage, often called hydatitis, is found in the liver and other visceral organs of humans and herbivorous animals, including sheep, goat, cattle, and horse. The adult worm measures about 3-6 millimeters in length. It consists of the scolex, often colored head and neck, and strobila. The scolex piriform with the foresuckers and rostrum bearing two circular rows of hooklets. The strobila is composed of three proglottids: mature, and gravid proglottids. The gravid proglottids contain a branched uterus filled with eggs. The lifespan of the adult worm is 30 months. The eggs Echinococcosis is indistinguishable for those tiny species. The larval form hydatitis develop in various organs of the intermediate host at the site deposition. The embryo slowly develops into the hollow bladder and cyst filled with fluid. This becomes the hydatitis. It enlarged slowly and reached a diameter or 0.5 and 1 centimeters in about 6 months. The growing cyst evokes a host tissue reaction, leading to the formation of fibrous capsule around it. The cyst wall secreted by the embryo consists of 3 layers: pericyst, ectocyst, and endocyst. The cyst is filled with fluid which antigenic, and its release into the circulation may cause anaphylaxis. Anaphylaxis forms the germinal layers of the endocyst. The small knobs protrude into the lumen of the cyst. This enlarge becomes vacuolated and are filled with fluid. These are brood capsules which are initially attached to the germinal layers by sulcus, but later detached and released into the fluid-filled cyst cavity. From the inner walls, brood capsules protosclerosis develop inside a major hydatid cyst. Was a generation of cyst. Daughter cyst and granddaughter cysts may develop. Cysts contain hydatid sand which made of protosclerosis and hooklets. The cyst takes many years to grow into the large cyst, causing clinical signs is the life cycle of Echinococcosis granulosus. Sensilator complex is the adult worm. Adult worm resides in the small intestine of the definitive host. Gravid proglottids release eggs that are passed in feces and immediately infectious. After ingestion of the suitable intermediate host, eggs hatch in the small intestine and release 6 hooked encospheres that penetrate the intestinal wall and migrate through the circulatory system into the various organs, especially the liver and the lungs. In these organs, the encospheres develop into the zig-walled hydatid cysts. The enlarges gradually, producing protosclerosis and daughter cysts that fill the cyst interior. The definitive host becomes infected by ingesting the cyst-containing organs of the infected intermediate host. After ingestion, the protosclerosis evaginate attached to the intestinal mucosa and develop into adult stages in 32 and 80 days, humans are aberrant-method hosts and become infected by ingesting eggs. Oncospheres are released in the intestine and hide that developing various of organs if cysts rupture, and the liberated prostoscolis might create secondary cysts in other sites within the body, often called secondary echinococcosis. Secondary echinococcosis. Epidemiology. Epidemiology, the most common models of transmission to humans is by accidental consumption of soil, water, or food that has been contaminated by the fecal matter of an infected dog. Echinococcosis eggs that have been deposited in soil can stay viable for up 2 years. The disease most common found in people involved raising sheep. As a result of the sheep role as an intermediate host of the parasite in the presence working dogs that can eat all offal infected sheep in areas of endemicity. Annual cystic echinococcosis incidence ranges from less 1 to 200 per or to 100,000 is pathogenesis is related to the deposition of the hydatid cystis, larval form of parasite in various organs. Hydatid cyst is the fully developed hydatid cystis of Echinococcosis granulosus, unilocular and subspecifical in shape and size. Various form is a few millimeters to more than 30 centimeters, usually size 5 to 8 centimeters. It appears as filled bladder-like cyst forms. Cyst wall consists of 3 layers: the pericyst, ectocyst, and the endocyst. Outer layer hosts the derived consists of fibrous tissue and blood vessels produced by the host cellular reaction. Ectocyst is the middle layer of the parasite derived. It is a tow elastic, glycan-rich cellular hyaline layer. The variable thickness also measures about 1 millimeters. It resembles the white and hard-boiled egg. Endocyte is the inner layer. Parasite derived germinal layer measures about 22-25 nanometers thickness. It consists of number of nuclei embedded in protoplasmic mass. Hydrated fluid is clear, colorless to plate, yellow. It has a pH 6.7 and a specific gravity of 1.005 to 1.010. Chemical composition is the sodium chloride, sodium sulfate, sodium phosphate, and succinators. It is antigenic, toxic, and anaphylactic. Broad capsules arise from the inner side of the endocyst and contains number of protoscolices which are hit. Hydrated sand is a leaking fluid from the cyst. Pathogenesis is a variety. Hydatid cyst, primary cyst, is formed directly from the oncosphere, released from the eggs ingested. Secondary cyst is formed due to the breakage of the primary cyst by trauma. The secondary cyst is carried in the circulations to various organs. As a follow cyst is a cyst without broad capsules and protoscolices, and genous daughter formed by the breakage of the broad capsule into the hydatid fluid surrounded by ectocytes and endocytes. The fate of the hydatid cyst is spontaneous resolution might happen to few cysts. Rupture of the cyst may either to lead to formation of the secondary cysts. Anaphylactic reactions to the hydatid fluid, antigens. Infection usually occurs in child but gets manifested in adult life. Site is the most common site of the location of cystic liver, about 60-70% right lobe, and the lung about 20-30% may be found in any organs like spleen and kidneys, 3-5%, brain and heart, 1.5%, and rarely bones. They grow up to 5-10 centimeters in size within the first year and can survive for years and even decades. As asymptomatic many cases as asymptomatic infection is detected only incidentally by imaging studies. Clinical features sometimes occurs due to pressure effect of the enlarging cyst leads to palpable abdominal mass and hepatomegaly, abdominal tenderness, portal hypertension, and ascites. Obstruction is the daughter cyst may erode into the biliary tree or bronchus and enter into the lumen to cause cholestasis and dyspnea. Secondary bacterial infection can cause pyogenic abscess formation in the hydatid cyst. Anaphylactic reactions to the cyst leakage or rupture may be associated with severe allergic reactions to hydatid fluid antigens leading to hypertension, syncope, and fever. Outcome of the disease depends on the size and location. Young children are more associated with extrahepatic cysts in the lung and the brain and orbital situs. Orbital situs is the clinical features. The liver is the most common site of the Echinococcosis, the pastoral strains, about 60%, followed by the lungs, 25%, and the cystis is seen less frequently in the spleen, kidneys, heart, and bone, and the central nervous system. Is the picture is the boy with abdominal distension due to cystic echinococcosis of the liver, as shown by ultrasound imaging. Laboratory diagnosis. Diagnosis of the echinococcosis relies mainly on findings by the ultrasonography or other imaging techniques supported by positive serological tests. Is in seronegative patients with hepatic image findings compatible with the echinococcosis. Ultrasound guided find needle biopsy may be useful for confirmation of diagnosis. During such as procedures, precaution must be taken to control allergic reactions and prevent secondary recurrence. The event leakage and of hydrated fluid of the protoscolysis. Protoscolysis skin test. It is a immediate hypersensitivity reaction to the hydrated fluid antigens developed by Kazoni in 1911. Antibody detection. The screening tests various antibody detection methods are evaluated using cryogenic granules, cystic fluid antigen. They show variable results, about 60-90% sensitivity. Antibody detection is the immunodiagnostic tests can be very helpful for the diagnosis of echinococcal disease, particularly in conjunction with imaging, and should be used for invasive methods. Indirect hemagglutination, indirect fluorescent antibody tests, and the enzyme immunoassays are sensitive tests for detecting antibodies in serum of patients with cystic disease. Sensitivity rates vary from 60 to 90%. 90% at present. The best available serological diagnosis is obtained by using a combination of tests. Indirect hemagglutination and enzyme immunoassays can be used for screening. Positive reactions should be confirmed by immunoblot assay. Immunoblot assay by antigen detection with the ELISA, counter-current immunoelectrophoresis, and latex agglutination tests are available to detect specific antigens in serum and urine. Imaging methods play an important role as they are non-invasive methods which can detect the cyst incidentally in asymptomatic individuals, and in seronegative cases, X-rays detect cavity and calcified cysts and cysts in the lungs. Ultrasound detects both single and multiple cyst inhalations. Computed tomography scan can detect 90 and 100% of cases. It detects more accurately the number, location of the cyst and the complications. Histological examination shows surgical removal cysts can be subjected to histopathological strains like James, hematoxylin, and eosin, and periodic HCG strains, and to demonstrate the three layers of cyst wall. Molecular metastasis PSI targeting mitochondrial DNA has been developed. The pictures show it hidden cysts in the liver of an 11 years of girl. Hidden cysts measures 6-7 centimeters in diameter and arose in the lung of the first patient detected by X-ray methods. Hidden cysts in the liver. The patient had 3 cysts marked by arrows in the right lobe. 2 were completely calcified, and the large one in the background was partially calcified. The picture shows the cross sections of inorganic roses strained with hematoxylin eosin host tissue and encapsulated of the hided cyst wall, which is composed of an acellular laminated layer and nucleated geminal layer, from which the blood capsule arises. Inside the blood capsule are numerous protoscolices with visible hooklets. Magnified beef of the Echinococcosis granulosus protoscolices stained with hematoxylin eosin. Eosin is not the row of hooklets. Prevention and control is prevent dogs from eating animal carcass or offal, particularly deworming of dogs. Personal hygiene. For treatment, we use puncture, aspiration, injection, and respiration methods is carried out in the early stages of disease. This method uses in Western countries. Surgical treatment in cases where the cysts are accessible. Patients with small multiple cysts can be treated with albendazole. Praziquantel may be useful prior to surgery, and cases or spillage of cyst contains during surgery. The next disease is alveolar echinococcosis, the causative agent of Echinococcus multilecularis. Echinococcosis alveolar echinococcosis is a zoonotic disease caused by tapeworm. Echinococcosis multilecularis is recognized as an important emerging parasitic disease and recently ranked as third most relevant foodborne parasitic zoonosis. Alveolar echinococcosis is a very serious disease caused by the metastatoid stage of Echinococcus multilecularis and is characterized by tumor-like infiltrative growth. Larval stages are often called metastastoda, colored multilocular or alveolar hyded cyst. Common name multilocular hyded worm, fox tapeworm, alveolar echinococcosis, also known as alveolar hyded disease. In humans, metastatic cells of Echinococcosis multilecularis proliferate within the liver, inducing slowly progressive life-threatening tumor life growth. Cause foxes, coyotes, domestic dogs, and other canines are defined hosts for the adult stage of the parasite. Cats may also be involved with rodents such as mice, serve as intermediate host. Humans can become an aberrant intermediate host by accidentally ingesting eggs of Echinococcosis multiloculis when handling infected animals or consuming contaminated food, vegetables, and water, except in rare cases where infected humans are eaten by canines. Humans did and as incidental causes, an aberrant intermediate host that doesn't allow transmission to the defined host. For the Echinococcosis multiloculis hosts, the organisms Echinococcosis multiloculis defined hosts foxes, dogs, the canida, and the cats. Intermediate hosts, small rodents. Geographical distribution: Echinococcosis, alveolar echinococcosis is found worldwide, mostly in northern latitudes. Cases have been reported in Central Europe, Russia, China, Central Asia, Japan, and North America. However, since 1990s, the prevalence of the disease seems to be increasing in Europe, not only in the historical endemical areas, but the neighboring regions. Transmission is alveolar echinococcosis usually occurs in wildlife cycle between foxes and other carnivores in small mammals, mostly rodents, acting as intermediate hosts. Domesticated dogs and cats can also act as definitive hosts. Habitat is adult worm is smaller than Echinococcosis granulosa and lives in the intestine of the foxes and dogs, which are the definitive hosts. Larval stages is found in the liver mammalian species, including man. The adult stages is measures about small than Echinococcosis multiloculis infection. Multiloculis is they have hooks. They have hooks and uterus. They contain three parts: scoliosis, immature proglotis, and gravid proglottitis. The life cycle of Echinococcosis multiloculis. The adult stages lives in small intestine. They measures about 1.2 4.5 millimeters long and resides in small intestine of the definitive host. Gravid proglottitis release eggs that are passed in the feces are immediately infectious. After ingestion by the suitable intermediate host, eggs hatch in the small intestine and releases 6 hooked anchospheres that penetrate the intestinal wall and migrate through the circulatory system into various organs, primarily the liver. The anchospheres develop into the multi-chambered, multilocular, thin-walled alveolar hydatid cysts that proliferates by the successive hot water budding. Numerous protoscolosis develop within the cysts. The definitive hosts become infected by ingesting the cyst containing organs of the infected intermediate host. After ingestion, the protoscolosis evaginate, attach to the intestinal mucosa, and develop into adult stages in 32 and 18 days. Humans are aberrant intermediate hosts and become infected by ingesting eggs. Oncospheres are released in the intestine and the cysts develop within the liver. Metastasis is a dissemination to the other organs, lung, brain, heart, bone may occur if protoscolosis are released from cysts, sometimes called secondary echinococcosis. Epidemiology: the incidence of the human infestation with Echinococcosis multilecularis is a disease increasing in urban areas as with foxes. Foxes is an important reservoir species of the Salvatic cycle migrating to urban and suburban areas and gaining closer contact with human populations. Also, restricting fox enclosures for the hunting with the infected animals spreads the disease. Children, healthcare workers, and domestic animals are at risk ingesting the eggs of coming into the contact with the faces and infected wild foxes. Alveolar echinococcosis is endemic in Central Asia, including Kazakhstan. In arid regions of Kazakhstan, characterized by desert and semi-desert landscape, forky of echinococcosis. Alveolar echinococcosis is rodents associated with water and aggregators around oases in Kazakhstan. The presence parasite is more widely distributed in mountainous and steppe habitats. Pathogenesis and the clinical features. The stimulate malignant neoplasm, the cirrhosis, numerous small irregular cysts, all less than 2 centimeters, border with union involved, liver appears infiltrative, has a thin and laminated wall, germinal layers is absent and very attenuated, forms small vesicles that invade adjacent structures, result in an infiltrative multilocular fibrotic maze that has a capability of the metastasizing. Inner portion may be necrotic, calcification in 70%, broad capillus protoscores seen in less 10%. 10% is the symptoms of the alveolar echinococcosis vary according to the site of infestation. For example, liver, lung, brain, and bone. Symptoms of the hepatic alveolar echinococcosis principally cholestatic jaundice and epigastric pain. Invasion of the bile ducts of the blood vessels leads to several complications such as cholangitis, portal hypertension, and biliary cirrhosis. Portal hypertension, the secondary biliary cirrhosis are most frequently observed clinical features. The disease can progress to the cirrhotic stages is after long lesion to asymptomatic periods. The clinical symptoms of lungs involvement in the alveolar echinococcosis are hemoptysis and the chest pain, cough with expectoration and exertination dyspnea. If brain involvement occurs, increasing intracranial pressure, epilepsy, and neurologic disturbances such as dysarthria, hemiparesis, and cranial nerve pulses may be observed. At gross pathologic analysis, hepatic lesions in alveolar echinococcosis appears as an infiltrative multivascular mass composed many cysts, irregular shape in size, diameter measures about less 1 to 30 millimeters with associated fibrosis and calcification because alveolar echinococcosis is caused by tumor-like and cyst-like tapeworm larvae growing in the body. The pictures shown is echinococcosis multilecularis in liver tissue stained with chemotoctilin and eosin. Higher magnifications about 400 of the specimens in the figure notices the pair of the reflectors hooks, yellow arrows, and so the hooks do not stain with the hematoxylin eosin but may be visible with the proper adjustment of the microscope. So for diagnosis, we use imaging techniques such as computer tomography scans, ultrasonography, and magnetic resonance imaging sensors are used to detect the cystis after a cystis has been detected, and the serologic tests may be used to confirm the diagnosis. Alveolar echinococcosis is typically found in older people, and older people for prevention and control, for control don't touch foxes and covered and wild animals. With the carnivores, don't keep wild animals, especially with the canines. Don't allow your dogs and cats to wander freely or to capture and eat rodents. If you think that your pet may be eating rodents, consult your veterinarian about possible preventive treatments. Wash your hands with soap and warm water after handling dogs and cats and before handling food. Teach children the important washing hands and to prevent infection. Do not collect and eat wild fruits and vegetables. Pick it directly from the ground. All picked foods should be washed carefully and cooked eating before eating. For treatment we use some surgical methods. Surgical surgery is indicated if lesion can be excised with safety margins. Long-term antiparasitic treatment with albendazole is mandatory. Preventive measures include the avoidance of contact with horses' faces, use consequential impregnated betas, regular treatment of dogs or cats with consequential, as well as hand washing and improvement hygiene. Is uses references. Thank you for your attention.